today. We're truly, truly blessed. So first, I'd like to introduce T. Ashizawa, who received his medical degree from Keio University of Medicine in Tokyo. After moving to the US, he completed his neurology residency and muscular dystrophy association membership fellowships in neurochemistry and clinical neuromuscular disorders at Baylor College of Medicine. He's a board certified in neurology by the American Board of Psychiatry and Neurology. He serves on multiple grant review panels, including NIH study sections, and on the scientific advisory boards of the Association Française contre les Myopathy, and previously served on the advisory boards of the Myotonic Dystrophy Foundation, as well as our own advisory board, the National Ataxia Foundation. As a physician scientist, some of Dr. Ashizawa's current research is funded by the National Institutes of Health, the Muscular Dystrophy Association, and the Myotonic Dystrophy Association. He is studying neurogenetic disorders caused by expansions in DNA sequences that are connected end-to-end and are abnormally repeated, such as myotonic dystrophy, spinocerebellar ataxias, Friedreich's ataxia, and Huntington's disease. He'll be our first speaker. Our second speaker today is Dr. Sophie Tenzinas de Monticello, who is a faculty member at Sorbonne Université, where she is a member of the Paris Brain Institute. After completing her medical internship in public health, Dr. Tenzinas de Monticello completed a PhD in genetic epidemiology. Her area of expertise focuses on biostatistics and epidemiology in the area of neurodegenerative disease. Of note, she has developed several measures of disease scales, including the SARA and Composite Cerebellar Functional Severity Score. Dr. Tenzinas de Monticello analyzes the usefulness of these scales in natural history studies and clinical studies. And our third speaker today is going to be Dr. Gülin Öz, who is a professor in the Department of Radiology and the Center for Magnetic Resonance Research. Following her Bachelor of Science degrees in physics and chemistry and a PhD in biochemistry, she continued with a postdoctoral training at the CMRR, where she later joined the faculty. She uses high-field multinuclear magnetic resonance spectroscopy to explore neurochemical and metabolic alterations in disease that affect the brain, and has spearheaded the utilization of neurochemical profiles to assess cerebellar changes in patients with neurological diseases and their response to treatment. Dr. Oz focuses on the application of advanced MRS methods in neurodegenerative disease and diabetes. She further leads efforts to facilitate the use and standardization of robust MRS methodology in the clinical setting. So with that, it is my pleasure to turn over the floor to our first speaker, Dr. Ashizawa.
All right, thank you very much. Let's see. So my name is Tetsuo Ashizawa, that's my true name, but known by the nickname T. It's spelled T-E-E in the Ataxia research community. I am the contact principal investigator and one of the 5 principal investigators of the largest NIH-funded clinical study of SCA, spinocerebellar ataxia to date, known as REDI-SCA. So we're going to talk about a lot about the REDI-SCA. I'm going to describe what natural history studies are and what it is like to participate as a study subject and why they are important. Following my talk, Dr. Tenzinas de Monticello will talk about what we learned from REDI-SCA study and other studies, focusing on clinical data, and then Dr. Oz focusing on imaging biomarker studies. Next slide, please. What is natural history study? It is pre-planned, it's an important keyword, and observational study intended to track the course of the disease, in this case, SCAs, without interventions, namely no treatment involved in those studies. We will learn about what kind of patient populations are out there and how the disease progresses over time. What do patients think important with regards to their symptoms and signs of the disease? Because the FDA, Food and Drug Administration, they're the gatekeeper of the drug approval for treatment. They really put the emphasis on is whether the efficacy really matters to the patient's daily life. Then identified new measurable biological indicators other than patient symptoms and signs of ataxia. They're called biomarkers. And finally, statistical characteristics of different ataxia measurements. Next slide, please. So what is natural history and why is it important? Many people may say it is not a treatment trial, so who cares? But in rare diseases like SCAs, little is known about natural history. We know that most SCAs are progressive and eventually disabling, but we need to know how fast the progression is and in what way the disease progresses. Natural history data are critically important for designing optimal clinical trials. For example, we would like to include the population of patients most likely to respond to the proposed treatment. And we need to choose the study size, number of the participants in the clinical trials, and also duration of the clinical trials. And this is particularly so because we do not have the luxury of large population of patients to be studied in many clinical trials. So if we have 3 or 4 large phase 2 clinical trials simultaneously going on, then we run out of patients who can be recruited in those clinical trials. So we have to be choosy. We have to have the clinical trial designed perfectly. Next slide, please. So too many ataxia clinical trials have failed in the past to adequately test the efficacy of the therapeutic interventions due to poor trial designs. This was due to an inadequate understanding of natural history. So these poorly designed clinical trials may have ruled efficacious drugs ineffective and vice versa. So that's a detriment. Not only it's wasting time, but it's actually set the field back. Next slide, please. Who can participate in a natural history study and how? So first, identify an available natural history study for your disease. The NAF website is an important resource, and you can immediately identify NAF's natural history study ongoing currently. And look at the inclusion and exclusion criteria of the natural history study that you're interested in. The criteria for participation in the natural history study vary from study to study. While they may be broadly set to capture the wide range of patients with different characteristics, they may target the patient population with inclusion and exclusion criteria very similar to those of upcoming clinical trials. We need to know genetics, particularly in SCA. NAF's natural history study accepts SCA 1, 2, 3, 6, 7, 8, 10, and now I think SCA 27B, that's a new disease described this year with a GAA expansion in intron of the FGF14 gene. Now, age range of the study is important in the inclusion criteria, and then stage and the severity of the disease is also important. And ability of the participants to get into the study and actually provide the data, that is important. So after you see those things, you can contact the PI, principal investigator of the natural history study. Next slide, please. So what happens in natural history study? What would participants experience? So signing the informed consent is the first step. This is called the ICF informed consent form. And if you decide to participate, you can call the study site and the coordinator will go over with you what this study is all about, what the pros and cons, what the limitations and risks. Visiting the study site in person with regular intervals, like a 6 months or a year interval, is very important and required because we have to examine you. The virtual visit, AI-based remote monitoring, etc., are under development, but it's not quite ready yet, but in the future, it'll come. Completing clinical outcome measures, donating blood and CSF, having MRI and other tests, filling out questionnaires, etc., are the main things that you have to go through and participate. Now, again, I reiterate that natural history studies are not clinical trials of therapeutic interventions. So there is a limitation, but at the same time, it is important. Next slide, please. So why are natural history studies important? Why do you participate in despite of no treatments? So next slide, please. To design optimal clinical trials, we have to select clinical trial sites, and but this natural history study will identify those favorable sites in terms of patient populations, enough number of subjects, excellent study team with expertise, etc. And making inclusion and exclusion criteria is based on natural history study. Sample size and duration of the trial is also based on the natural history study, the progression rate, and also on how long we can do it, and the feasibility of the clinical trial sites. Then clinical outcome measures to be used. This is an important point. I mean, natural history study, we use certain predetermined, pre-planned clinical outcome measures, as well as biomarkers. But actually, looking at the data, we can tweak those things to optimize to be used in the actual clinical trials. And selection of the clinical trial design is very important. Now, we have a limited number of study subjects that you can enroll. So even if everybody is very enthused, the sample size, quote unquote, is limited. So statistics is compromised compared with a common disease like heart attack, stroke, high blood pressure, etc., etc. Again, it is important to emphasize that these are essential data that in natural history data, from natural history data, to determine the feasibility of clinical trials. Next slide, please. So we have just finished all scheduled clinical visits of the study subjects in ReadyScar about a month ago. However, the visits for MR scanning is ongoing until the end of this August. And our participants did not directly benefit from the study in ReadyScar, but the study identified the large enough target study population and provided important missing pieces of natural history of these diseases. And we recognized needs for sensitive clinical assessment measures, and those are reproducible and reliable, and which are in the process of improvements. For example, SARA has been used. SARA is a scale for assessment and rating of ataxia developed by Dr. Thomas Kroggetter and his group in Germany. And this has been used for a number of years as a gold standard. However, after doing the natural history study, we realized that may not be the optimal. We need to tweak that to improve the characteristics of clinical outcome assessment measures suitable for and optimize the clinical trials. And then we learned a lot of things about development of useful biomarkers, and following two speakers will touch on those things. And finally, I think if we have time, Dr. Tesnas Dumont-Sell may talk about clinical trial designs, which is suitable for the small population like ICA sufferers. So this is the end of my talk. And here on, Dr. Tesnas Dumont-Sell and the odds will take over. I think we will have a discussion and question and answer session at the end. Thank you. Sophie, please take over.
Next slide, but I think the next is still for you.
Oh, is this for me?
Yeah.
Okay, that's fine. Now, I already mentioned most of the stuff. So we have identified by ReadyScar, and ReadyScar is only focusing on SCA1 and SCA3. The SCA1, because SCA1 is the fastest progressive SCA among common SCAs, and SCA3 because it's the most common SCA worldwide. And we identified large enough target population, and we defined the natural history, and we identified that sensitive clinical assessment measures and with factors that need to be improved. And we identified useful biomarkers and the innovative clinical trial designs will be talked about. So without these, sound clinical trials cannot be designed. And the FDA demands these data before approving investigational drugs for clinical trials. So to obtain them, we need participation from eligible individuals in clinical trial readiness studies. So you have already that part of the natural history available in ReadyScar database. And if you participate in clinical trials, you are the fresh patient for that trial, and you have already trajectory of the progression. So that's a huge advantage. The data that you provide may help you and your relatives for participating in future clinical trials for the progressing trials. All right, thank you. Now I think I've ended.
Thank you, T. What did we learn from ReadyScar? I will present today the results from the baseline analysis, baseline data of the ReadyScar, which was published early this year. Next, Celeste. So this is some data we have from the paper. On the table, you can see that we included both patients at the ataxic and pre-ataxic stage of the disease, but we also included controls. And this is really important to differentiate symptoms that patient can observe, but that they are also present in controls. You can see that some controls had cramps, for instance, and they had cramps as often, even a little bit lower than pre-ataxic or ataxic. And then we observed what we already know, that ataxic patients have a lot of different symptoms, but we also have seen that pre-ataxic patients, which means that is subject here with SCA1 expansion, but without ataxic symptoms, they already have some mild symptom. Here for the SCA1 patient, we observed that these patients had already hyperreflexia. On the next slide, you have the result for both SCA1 and SCA3. Next, please. Yeah, you have the same graph on the left for SCA1, and you have on the right for SCA3. And you see that the picture is a little bit different with a higher proportion of patients with SCA3 we have diplopia, at the ataxic stage, but also at the pre-ataxic stage, and that the SCAD3 pre-ataxic patients also had often impaired vibration sense at ankle, which is a symptom monitored by the clinician. So thanks to the patients and control that were included in the study, we were able to show that at the pre-ataxic stage, the patients had already extracerebral sign, and that these extracerebral signs were different according to the genotype. Next, Celeste. So we also wanted to look for the disease duration. So in that analysis, we were able, for the subjects that are at the pre-ataxic stage, to estimate the disease onsets when the disease will, when the ataxia will start, and to have a disease duration for both the pre-ataxic and ataxic patients. And we were able to show that the severity of the disease increases with disease duration, which was expected. And we showed that on several clinical scales, the SARA, T already mentioned, the CCFS, which is a task that is performed with the hands. Also, there was a relationship with the cognition on this disease, but we didn't show a correlation with depression and fatigue, which are symptoms that the patient reports, but it's not linked to the disease duration, at least in our study. We have shown also that there is a link with quality of life and with activity daily living, which are affected more as the disease duration continues. So this is clinical outcome. And we also were able to look for a biomarker. Here is the NFL, which is monitored from the blood. And we have shown that the biomarker was increased with the disease duration, which allowed to monitor, for instance, in a clinical trial, this biomarker. Next slide. These results are based on the cross-section, the baseline analysis. We will have in September the data from the REDISCA for the longitudinal evolution. But I wanted to mention what we have done as analysis on the European cohorts that were done several years ago in Europe. On the left, you have the results for the ataxia patients, at the ataxic stage, where we were able to show the progression during the study with sophisticated statistical model. And on the middle, you have a graph. We looked for the SARA score, but on subjects that were included at the pre-ataxic stage. They were carrier, SCA1, 2, 3 carrier of the mutation, but didn't expressed already. And we were able to show the progression. Here I show you for the SARA, but we had several different outcomes that were measured. And this allowed to look to the different progression. You can see that the SCA1 patient progressed faster than SCA2 and SCA3. So with longer follow-up, we can learn more than just having cross-sectional. We can objectify the disease evolution. What I don't show you on this slide because it was too heavy, but we also find predictor of disease evolution. For instance, some genotypes, some based on the age at inclusion or some sign can be predictor of disease evolution. And we can then have potential predictive model to optimize the selection in a clinical trial, not only having the good design and the good number of patients included, but also try to select the patient that have the best chance to be responder for the trial. So now I let the microphone to Gülin.
Thank you, Sophie. Let me share my slides. All right. Well, hi, everyone. Thank you for joining us. And I'm joining you from Minneapolis, Minnesota, and I'm going to be talking about imaging and imaging biomarkers that we collect in the REDISCA study that you already heard about from doctors Ashizawa and Tezenes-Dumonchal. So hopefully after the short presentation, MRI is not a black box anymore, and you have a better idea of the utility, how we use this imaging technique non-invasively to monitor disease in the brain. So the question is, why participate in an imaging study, continuing on the natural history, the benefits of natural history studies topic? First of all, MRI, magnetic resonance imaging, opens a non-invasive window into the brain. You can not only evaluate anatomy, anatomical changes, shrinkage of the brain tissue, for example, of the cerebellum, but also you can study networks in the brain, how different parts of your brain are connected to each other and communicate with each other. You can evaluate chemistry, you can evaluate blood flow, function, etc. So MRI is not just one modality that gives you pictures of the brain, but it actually provides a wealth of information about function of the brain from different perspectives. So a number of studies have shown that magnetic resonance imaging with the different modalities is more sensitive to progression of disease than clinical assessments. But this was more limited to single site studies. So it was really important in a study like REDISCA to measure anatomy, chemistry, networks in the multi-site setting that would be used in a future clinical trial and assess if the sensitivity in this type of multi-site setting remains. We also are very interested in looking at changes in the brain before the onset of the motor symptoms, before the onset of ataxia, so that we can have this window into the brain before even the motor symptoms appear, so that we have a chance to potentially intervene in the future if there are detectable changes. And there were a lot of studies indicating the sensitivity, but again, it was important to check this, evaluate the sensitivity in the clinical trial setting with multiple institutions involved in the US and Europe. And then the last point is MRI may be more sensitive to detect treatment effects in clinical trials. This is still to be evaluated, and the potential there is that we may be able to reduce the number of participants needed to understand drug effects. So I need to quickly move my, okay. So I'll talk about the type of imaging we do in REDISCA. We use the standard MRI scanners that you're familiar with from the hospitals. And just a little bit of background, these MRI scanners we refer to as 1.5 Tesla, 3 Tesla, 7 Tesla, etc. And the Tesla is the unit that defines the strength of the magnetic field. So a 7 Tesla scanner has a stronger magnetic field that it utilizes than a 3 Tesla magnetic field. And clinical MRI scanners for a long time have operated at 0.5 to 1.5 Tesla strength, although now 3 Tesla scanners are more commonly available even in hospitals. But for this study, we chose to use the higher field, the 3 Tesla scanners, to have better information, more sensitive information, clearer images, more detailed information. You can think about it like the old CRT televisions versus the new high definition televisions that give you much better resolution. So that's what the higher magnetic field gives you. So that's what we have been doing in REDISCA, using these higher end scanners to monitor the brain. My slides are not advancing, I'm sorry. Okay, there we go. So, and we do look at anatomy, the structural changes in the brain, but we also look at connectivity, like this depiction shows in the corner here. We look at networks in the brain and how they are affected at different stages, different early stages of the disease. And we look at chemistry, and the data in that case looks like this. This is a spectrum, what we call a spectrum from a selected brain region like this. This is one individual, a healthy control volunteer. And on the right, I have the spectrum collected from comparable brain region from an individual with SCA1. And just looking at the patterns of these peaks, you can see, for example, this chemical, this is representative of one chemical and its concentration in this brain tissue, it's lower. So this happens to be a chemical that goes down with dysfunction in nerve cells. When the nerve cells are struggling, getting sick, this chemical goes down. And we can, in a single spectrum, see that even visually. So it's powerful information. So just like Sophie explained with the baseline clinical data, we have just published at the end of last year the results from the imaging component of REDISCA, the baseline, the first cross-sectional first visit results of imaging. And what we have seen is that changes in the brain are indeed detected by imaging before the onset of ataxia symptoms in this multi-site clinical trial setting. We had 6 imaging sites, 4 of them in the US, 2 of them in Europe, and we did all the analyses in a blinded fashion without knowing which data set is coming from which participant. And then Sophie and her team analyzed the data statistically, and we found indeed at the pre-ataxic stage, we are seeing some changes. So on this figure, I'm showing some example data from participants with SCA1 and participants with SCA3. And the color code here shows the differences we were able to reliably detect relative to control participants. The top row is differences during ataxic stage relative to controls. The bottom row is differences at the pre-ataxic stage. So these individuals have SARA scores of less than 3. They don't have the ataxia signs and symptoms yet, but we are able to detect, for example, in SCA3, you see these colored regions are affected in their connectivities in the brain. So then Dr. Ashizawa emphasized the importance that FDA is now putting on patient-reported outcomes. Anything we measure by biomarkers non-invasively need to be relevant to the patient's experience. So we collect these surveys that are called activities of daily living that patients report to us, how they feel, how they function, and then we look at their associations with the markers that we measure in the brain. And I have this one marker here. This represents the connectivity in the brain in one region. And on the right, I have a chemical marker in this region called the pons in the brainstem. And these plots show that they are strongly associated with our patients' experiences, what they reported in these activities of daily living. And that's why the title of our paper is Clinically Meaningful Outcomes that are collected by MRI. And then these figures show that these advanced imaging methods that we have been using at high magnetic fields are indeed quite sensitive to early changes in the brain. So on the left in SCA1, I'm showing a chemical measure, and these are data points collected. It is the concentration of this chemical we measure in the brainstem non-invasively. So this is not structure, it's chemistry. And these are the values we collected from control participants, and these are the values collected from pre-ataxic participants with SCA1, and you can see there's barely any overlap. And on the right, similar type of plot looking at networks. So these measures, these non-standard measures we found are more sensitive than even the structural MRI measures in the study. So how can we use this information? Well, we may be able to start future therapies as soon as these changes in the brain are detectable, even before the onset of the ataxic symptoms. And then we may be able to select trial participants who are closer to ataxia onset. T and Sophie already talked about this, and imaging may contribute to that participant selection for future trials as well. So how can you get involved in a natural history study? NAF has a wonderful website. So you go to their website and click on the Participate in Ataxia Research button. You come to a page that looks like this with some options. Then you select the research studies at the bottom here, and then you get a list of interventional studies, observational studies, etc. So I have a screenshot here from the observational clinical trials section. And you can see the name of the study, the condition the study focuses on, their location, and their age criteria. And then you can go to the study's specific information page and see if it fits your interests, your condition, and if you may want to participate. Then there is this paragraph here about the longest running ataxia observational study that NAF has been sponsoring. And to learn more about it, you click here to learn more. And then you come to a page that talks about the CRC-SCA Natural History Study, Clinical Research Consortium for the Study of Cerebral Ataxia. This is a study that has, I believe, up to 20 sites, institutions in the US where you can participate. So far, it has been a study focusing on clinical outcomes, but we are working on adding an imaging component for this study too. And it says, it describes, this is a natural history study and biomarker identification to accelerate treatment development. And then here's a map that shows the locations of the participating sites. The criteria for this particular study is participants who have confirmed diagnosis of SCA 1, 2, 3, 6, 7, 8, or 10. So, and it's a longitudinal follow-up study, like you heard from Dr. Ashizawa in the first presentation. So thank you. And I have this website here about the natural history studies from NAF. I encourage you to take a look there, and thank you so much for joining us. I'm going to stop sharing so I can see everyone else. And we look forward to your questions.
Thank you three so much for a fantastic presentation and an overview of natural history studies and the findings of ReadySCA. So we're going to jump into questions in a second, but just a reminder for all attendees, if you do have a question, to please put it in the Q&A window at the bottom of your screen. If you click that, you can type it out and it will be sent directly to me so that I can ask it on your behalf. Unfortunately, we can't unmute attendees. So if you're raising your hand, we won't be able to call on you. So if you have a question, please type it in that Q&A window. And we already have some of our first questions coming through. And the first one is for Dr. Ashizawa. So we have an attendee here who has heard of something called a prospective study. Could you explain what that is and if that's different from a natural history study?
Okay, so prospective study is similar to pre-planned. I mentioned that. So you make a plan, what kind of things will be obtained as data, okay, as opposed to retrospective study that is based on the chart review. But medical chart is not documented according to a certain set of predetermined, pre-planned parameters. So that is a prospective, but if you think about clinical trials, all clinical trials are prospective. Unless there's a retrospective review of the therapeutics, that's a retrospective study. But most of the clinical trials soliciting enrollment is a prospective study. So it's from here, pre-planned and foregoing into the future. That's prospective.
Fantastic. So it's a word that researchers and clinicians use to describe the timeline if it's prepared ahead of time, if they're looking back in time. That's very good to know. We have another question for you, Dr. Ashizawa. Can someone enter a natural history study if they are being treated by a symptomatic relief drug, or does that depend on different natural history study criteria?
If it's documented, they should be, depending on the natural history study criteria of inclusion and exclusion. But most of the time, I think we try to be inclusive. Really, Sky is a little bit more restricted. For example, physical therapy cannot be changed because it's being demonstrated. Physical therapy is efficacious to decrease the level of ataxia. So if you change that, we cannot say this is a true natural history or it is contaminated by the efficaciousness of the physical therapy.
That's very helpful to know. Our next question is from Dr. Tenzinatsu Monticel's section. So you talked about in your graphs that certain people were pre-ataxic. Could you explain what this means? Do they have the gene? Where are they? Are they not experiencing symptoms yet? Could you elaborate on this, please?
Yeah, indeed, it's people that have the gene but do not express cerebral symptom ataxia. They don't have ataxia, and in the study we define by the SARA score, and a SARA score of below 3 is mentioned as pre-ataxic. Why this cutoff is because we have observed in controls that people can have a SARA score of 0.5 to 2.5, and then we classify these patients as pre-ataxic.
That's very helpful to know in terms of how folks are processed and put into different groups in order to participate in these studies.
And for us, it's meaning that they are early subject. They have the gene, but they are not really, they have really symptoms that are mild and that they are not ataxia.
That's it. That's good to know. Our next question is for Dr. Oz's section. So you were talking about the different scans and tests that people do to participate in natural history studies. Is it possible for the researchers to share individual findings with patients, and how does that work?
Yeah, it is possible. So before we start any study, we get ethical approval from institutional review boards that review the protocol, what we will be able to share with participants or not. So everything gets reviewed in detail. And if the study has that process in the protocol at the beginning, we can share the results. So for imaging results, it's our general practice to have that option open. So we do share imaging data, but we limit it to the structural imaging data that the physicians of our participants will be able to look at and interpret. Others require special processing methods, and they will not be meaningful to a neurologist in the office. So, there are some limitations, but that's open. It's an option. Yeah.
So this is something that patients would be informed of when they're signing that informed consent. What kind of information will be shared with them during this process?
Yes, and some studies, again, it depends on the study. Some studies will have sharing of results with everyone. Others will have sharing of results if the participant requests it, or their physician requests it. So it all depends on the specific consent form and the protocol.
Mm-hmm. Fantastic. Our next set of questions draw on all three of your experiences. So please feel free to jump in who would be best to answer the question. First, we have a question. So you mentioned throughout the presentation that natural history studies, you need to check in every so often to gather data, see how things progress over time. How long do most natural history studies last in terms of timescale?
That's okay, go ahead.
No, go ahead.
So red sky is probably fairly standard, but it's all contemplated for 5-year follow-up. Well, actually 3-year follow-up, but extended to 5 years. And it was complicated by pandemic of COVID-19. Natural history study arguably started back in 2009, and it's still ongoing. So it's a very long natural history study. So it all depends on, and a good resource to check that is a clinicaltrials.gov. That's just type in clinical trials, that's one word, .gov, and then return. And you have a screen for the search engine, and then you type in your SCA or whatever you type, and then natural history, then it'll show up.
Yeah. Yeah. Often the duration of the trial is defined by the funding. Us as a researcher, we would love having 10 years follow-up, but often the funding are not there, and the cohorts just disappear because there is no funding left, and that's a key issue for the duration of the study.
100%. This is very important work, but can also be quite costly and can be a barrier to ongoing research.
Yeah.
Then I'm just looking through. We have a ton of questions coming in. We'll try to get to as many of them as possible in the next 9 minutes. We have a number of people asking a similar question. They have ataxia, but they have an unknown type. They don't know the exact cause. For these folks, are there any options to participate in natural history studies?
I can answer something. I don't know in the US, but I know that there are different studies in Europe. There is one in Germany, there is one in France that are open to less frequent or even unknown form of ataxia. And I think that looking to the clinicaltrial.gov website give the information of if there is something somewhere in the world related to your SCA, your genotype, or even unknown genotypes.
Dr. Oz, did you want to add anything to that?
Yeah, maybe I'm not. So that if you are living in the United States or other countries, that there is no blanket natural history study like in Europe, I would strongly recommend and that person should go to Ataxia Center of Excellence of NAF and AF. And there are experts who are familiar with natural history studies. As a part of the clinical practice, they can see you, but they can document all relevant parameters. So we have symptoms, signs, and scales, SARA scales, and other studies, even MRI studies, just regular standard MRI, not as sophisticated as Dr. Öz was describing. But then that's going to be a very important resource when new natural history study come and focusing on certain SCAs. Then we can look at that and you can be genetically tested and enroll. Same thing can be said in future clinical trials. And if your phenotypes, I mean, manifestations, signs, and symptoms match with the SCA type of the clinical trials, you can be tested.
Yeah, and I wanted to add, in terms of therapeutic trials, if a medication targets the symptoms, the ataxia symptoms which you have, you may be able to participate even with unknown genotype.
And I think you led into the next question I was going to ask. We had a few people with rarer SCA types who are wondering, there's no natural history study for my ataxia type. How can I get involved? How can I participate in research? And I will echo Dr. Ashizawa's suggestion of getting in touch with an ataxia center of excellence near you. You can find a list of those on the National Ataxia Foundation website, and one of our people will drop that link in the chat momentarily. And then we have a specific question about the protocol for ReadySCA. This participant was interested, so you focused on SCA1 and SCA3, and then also for CRC-SCA, there's certain ataxia types that are included, there's others that aren't. They were wondering why SCA5 patients aren't being included in some of these particular studies, if there is a reason behind that.
So I may mention that the SCA5 is a slowly progressive disease and caused by spectrin 2 gene on chromosome 11. And this was discovered by Dr. Laura Raynham at the University of Florida, my close colleague. And I would suggest that you contact her, and also University of Florida will be, I assume, a center of excellence for the Ataxia Center of Excellence. And Dr. Subramani and Dr. Burns are the investigators and directing the Center of Excellence.
And to add to that, the reasons to focus on certain ataxias typically comes down to feasibility. Would we be able to reach the number of participants in the study to have meaningful statistical analyses? And in the case of ReadySCA, for example, we focused on SCA1 because it's the fastest progressing type of the most common ataxias, and SCA3 is the most common globally. But that is a start. That doesn't mean we won't strive to do the similar work with other SCAs. This was just a start, and within the funding limitations, we had to restrict it to 2 SCA types, but we are already planning to expand into other SCAs.
So it's going back to that funding component about how in an ideal world, we'd be able to do things faster and quicker, but there's only so many resources. So we're trying to do what we can as fast as we can within the constraints that exist.
And even when you focus on a few ataxia types, you do learn a lot, especially on running these multi-site studies. Can we collect these harmonized data, and how consistent they are between sites? Can we pull the information reliably? What is the variability? So, and for those questions, you do want to start with the ataxias where the largest cohorts are available, and then so you can expand to others.
So what I think there's an expression that a rising tide raises all boats. So if we learn from a certain ataxia type, then those information can be expanded upon and help other types. So that's very heartening to know. And then just looking through our questions in our final few minutes here, I think this one is directed at Dr. Ashizawa. Earlier in your portion of the talk, you mentioned that there's going to be studies coming down the pipeline for SCA27B, a fairly recent discovery, and genetic testing is becoming available for this ataxia type. Could you elaborate on this a little bit?
Yeah, so I just looked up in clinicaltrials.gov, and there is no specific study for SCA27B. Now, so 27B and 27A are different, two different things, but 27B is caused by GAA expansion in intron. That means it's not made into the protein. And that's very similar to Friedreich's ataxia. And as you know, Friedreich's ataxia has an FDA-approved therapeutic drug, such as omaveloxolone or SKYCLARY. And so that success is very encouraging, so that I think industry and investors and those people are interested in this disease. So that's my pure guess, personal guess, and that will be focused in the future.
And on that note, if as these trials are in development or about to be launched, as was mentioned earlier in this presentation, one of the things you often need in order to participate in clinical trials is genetic testing and a confirmation of ataxia type. We have updated information on testing for SCA27B available on the National Ataxia Foundation genetic testing page. We've been getting quite a number of questions just as it's very new and a bit difficult to navigate. So if you are interested in getting tested for SCA27B, there's updated information on our website. And with that, I'm just taking a look to see if we've missed anything, but it looks like we are coming to the end of our session today. So I'd just like to extend our deepest thanks and gratitude to our three speakers. So if everyone could join me at their own individual computer screens, giving them a small round of applause. So thank you for sharing your time and expertise with us today. We very much appreciate it. And thank you to everyone here who has attended and learned more about natural history studies. I hope you found this session beneficial and that it helps you understand things a little bit more. So with that, wherever you are in the world, good morning, good afternoon, good evening, have a wonderful rest of your day. Bye, everyone.